The study will try to use the circulating tumor cells, help with cancer diagnosis, prognosis, and even monitoring the therapeutic and response. And because previously and most of the diagnosis and prognosis is based on truly the tissue biopsy, we have to get the tissue sample and to do the diagnosis. But that's usually get the tissue, for example, the prostate, and is very time consuming, and it also can be a very painful procedure. So we can't just frequently take the tissue biopsies and to do that. But they'll use the blood sample activation. People get you to just blood test. That's easy to take the sample, and even we can to get the circulating tumor cells, and we can properly analyze them. So we can use them and frequently take the blood and just see during the treatment what's the genetic and all the biological features there. We can treat maybe more individualized and timely, and that's what is currently called the precision medicine is doing. Oh, that's also depending on which stage we take the sample. And for example, we doing the before the people during the diagnosis, and we just take to see if the circulating tumor cells are already there, and that may help with cancer diagnosis, and particularly can be even early diagnosis. Of course, my focus is mainly on work on prostate cancer diagnosis, which is always they can be diagnosed earlier. But for many other cancers, frequently early diagnosis is an issue that may help, and just take the blood to find the circulating tumor cells to help the early diagnosis. And also, but on the prostate cancer, we mainly work on the later stage disease, and just based on CTC to see how good the prognosis is. That's the current our work is on. And based on if there's more CTC, that means the cancer cells already get into the blood, spread to the body, and form the metastasis. That's more progressed. Usually that's maybe advanced disease, and there's a high mortality rate. So that's something we have to just stratify the treatment, who should treat heavily and who may not necessary. We found is also the CTCs, and that's particularly there's the different type of CTC. Previously, the CTC many people work on because the carcinoma is the epithelial cell type, and we worked on is we can detect the with the cell have transformed into the mesenchymal cell type. That is more linked to cancer metastasis process. We can detect those, and those linked to the cancer prognosis even more strongly than the epithelial type of the CTCs. That's what we are aiming to do. At first, we can, based on the one blood test, and to see if they got more of these circulating tumor cells. So we can just, based on this, we'll see they have a bad prognosis for prostate cancer, so we can treat them heavily. And for people, if there's prostate cancer, half of them, if the early stage and is not very aggressive, and they can survive more than 10 years, may not necessary to be treated and until they progressed. So that's something we just diagnose it and go to treatment. During the treatment, we can frequently take a blood sample that can be easily done, and then just monitoring the CTCs and see how they respond to the therapeutics. Should we change the therapeutics, or maybe even according to this, and during that time, we may have to think that we should stop the treatment, and because it's efficient, and until there's more tumor cells come, we treat them again. Yes, surely there's a cost issue. Is currently there's lots of the CTC isolation and system have been developed, and each of those systems is quite expensive. So we got with support from, we are in collaboration with Angle. They have the particular type of the CTC isolation platform, and so we got that donated by them. And it's not donated, or this is just support. We in the collaboration, they have to free rent of these machines, so we access that freely. And during the CTG analysis, there are some of the cost issue, but those costs are relatively much cheaper compared to lots of the other next generation sequencing analysis, which there is still cost issue. And another thing I have to say, we currently mainly look at how many circulating tumor cells, which is called the CTCs, and in the blood. And we did some also, there's a few biomarker, the protein marker and genetic marker in the CTCs. But in the long run, we may have to doing the whole genome or what's in the CTC, the genomics change and the whole transcriptome changes. That's something we are trying to develop as well. That will be much more expensive.